Hello, in this video we're going to be talking about cancer and the cell cycle. So let's not waste any of your time. Let's get right to it. Now when we're talking cancer and the cell cycle, we're talking about just a lack of controls. Okay, for something that's happened to where the control factors that we just spoke about in our previous video aren't working. Now most of us, if you've lived any time at all, you may have someone who's been affected by cancer, whose life has been changed by cancer, and so we're going to look at those processes. It comes down to the fact that the cell cycle controls, they're just not working. Something has changed with this particular cell, and they're no longer responding to all those things that are in place that we've spent 9 videos covering to control that. Now there are 2 kind of genes that help disturb the cell cycle when they get mutated. Something that's occurred that has changed them, and they make this cancer pathway a probability. That's tumor suppressor genes and proto-oncogenes. Now when it comes to tumor suppressor genes, these are genes on the chromosomes that have particular functions, but they're looking to control that cells that are out of control, and that's one of the environmental factors that could affect a cell going through cell cycle. That's just one of the many things that has to be checked to make sure. So an example of this is going to be P53. P53 is that negative regulator that we talked about, whose job it is to check out the DNA, to recruit things that are needed to fix the DNA, or make that decision to, hey, we're really in bad shape here. Let's just kill it off and to start from square zero, or the first square. But if that particular molecule gets mutated or it's not doing its job properly, then lots of bad things can happen, and we'll see that on this slide here. Now here's that P53. You can see it in the little blue box just to the left of the very first DNA strand that's being duplicated. If that P53 protein isn't working, okay, then cancer can develop, because that P53, it's designed to check, to fix, to recruit, help if needed, and if it's not working, then the needed checkpoints, they also will not be working. So the proto-oncogenes, again, they're normal genes who are trying to help prevent this or do the proper thing. But if they get mutated, then they become oncogenes, and they can become cancerous. And if you're going to have the 2 genes. And essentially now they're turned to the on position, and so therefore the cell will no longer respond to the growth factors or the checkpoints in place. The cell now just starts to divide through cell division without any checkpoints. So it's a situation where a cell is already bad, and it can become worse if it's not checking its own DNA and to go through that process. So here's a diagram that kind of just shows everything that we've been talking about. You get your proto-oncogenes, those growth factor receptors, anything that they're checking for or responding to will be there. You get your tumor suppressor genes, your RB proteins, and you can see the percents there that if those get mutated, or I should say in all cancer cases, the RB53 is mutated, or no, 50%, and just a traditional RB and 40%. So it's just super important that that molecule is taken care of and stays healthy. Well, that's our video over cancer in the cell cycle. It also brings us to an end for this video lecture series. The next time we come together, we're going to be looking at meiosis. We'll see you then.